Hello students, this is your Prasanna ma'am from NEET World. Welcome you all for a very interesting and interactive session of NEET previous year questions from the chapter Cell the Unit of Life. So if you look into the first question, which is recently asked in the year 2024, where he has given us the match the following. He is asking us to match the organelle and its function given in the list too. If you look into the nucleolus, nucleolus is what ma? Nucleolus is non-membrane bound organelle, which is, it is what ma? Non-membrane bound organelle present in nucleoplasm. Non-membrane bound organelle present in nucleoplasm. Okay, yes. So yes, this nucleus is involved in what ma? Site for active synthesis of RNA. This nucleolus will actively synthesize rRNA. tRNA and mRNA are synthesized in a nucleoplasm. tRNA and mRNA are synthesized where they are synthesized in nucleoplasm. Nucleoplasm. Clear? Next. So, A is option 3. Next, centrioles. Centrioles are the organization like cartwheel which forms the basal body of both cilia and flagella. What is the arrangement of cartwheel centriole has? That is 9 plus 0 arrangement, where there will be a central proteinaceous hub. To that 9 radial spokes are raised. To each radial spoke peripherally there are 3 doublets. Peripherally there are 3 doublets. Triplets, sorry. Peripherally there are 3 triplets which forms a centriole arrangement, that is 9 plus 0 cartwheel arrangement. So, B is option 2. Okay. Next, leucoplasts. Leucoplasts are the non-colored plastids which store the food material. If they are storing protein, you will call it as alluroplast. If they are storing starch, you will call it as amyloplast. If they are storing oil and fats, you will call it as elioplast. So, yes, leucoplast, what will be your option? Option 1. Next, Golgi apparatus. Golgi apparatus are the site of, Golgi apparatus will be the site of formation of glycolipids and glycoproteins. Glycolipids and glycoproteins. This function of Golgi apparatus has asked multiple times, so fix it in your mind. Golgi apparatus are the site of formation of glycolipids and glycoproteins. They are modifying the proteins and lipids which are synthesized from rough endoplasmic reticulum and smooth endoplasmic reticulum. So, Golgi apparatus, it is option 1. Leucoplast, it is option 4. So, now you tell me what is the correct order and choose the correct answer. 3, 2, 4 and 1. Option 1 is your answer. Next question beta. Next question. Here statement based question is given from this chapter. This we have already discussed in the statement based session. You can go through that also. Statement 1 he has given, like in bacteria the mesosomes are formed by extensions of plasma membrane. Obviously correct, wherein a bacterial plasma membrane will have an extensions either in the form of vesicles or in the form of tubules or in the form of lamellae towards the plasma membrane. This is towards what ma? Towards plasma membrane. Towards plasma membrane. So, yes, statement 1 is correct. Next, if you see statement 2, the mesosomes in bacteria help in both DNA replication, cell wall formation and cell division as well. They are also helpful in respiration and secretion. And what beta? Secretion. All these are the functions of mesosomes. So, yes, both statement 1 and statement 2 are correct. Your option will be option 1. Next question. If you look here, which of the following statements are correct with respect to Golgi he is asking, which is asked in the year 2023. It is important site for formation of glycoprotein and glycolipid, already discussed, right, in 2024 question. It produces cellular energy of ATP. Wrong. The cellular energy of ATP is produced by what ma? Mitochondria. It is produced by mitochondria, which is the powerhouse of the cell. It modifies the protein synthesized by ribosomes on ER. Correct. It modifies the proteins of rough endoplasmic reticulum. Ribosomes are on ER is nothing but what ma? He is speaking about rough endoplasmic reticulum. C is correct. It facilitates the transport of ions. Wrong. Golgi will not facilitate the transport of ions. It is indeed facilitated by plasma membrane. It is facilitated by plasma membrane. Next, it provides mechanical support. Wrong. Mechanical support is provided by cell wall of plant cell, fungal cell, okay, bacterial cell. So, what are the two options which are correct? A and C. So, your answer will be 4. Next question. If you look into it, which of the following are not considered as a part of endomembrane system? Endomembrane system includes what organelles beta? They include endoplasmic reticulum, Golgi, they include endoplasmic reticulum, Golgi apparatus, lysosomes, and vacuoles. Okay. They do not include mitochondria, which is double membrane bound organelle, chloroplast. These two are what? Semi-autonomous organelles. Semi-autonomous or self-replicating organelles. Semi-autonomous or self-replicating organelles. They don't include them. Next, Golgi complex is present. Peroxisomes are also absent. Lysosomes are present, not peroxisomes. So, which are not considered as a part of endomembrane system, then our answer will be A, C and E. Option 1 is our answer. Next question. If you look into it, if the pH of the lysosome, application question beta, focus on it, in 2022 it is asked. If the pH of lysosome is increased to alkaline, then what will be your outcome? What will be your outcome? Usually the pH of lysosome is less. pH of lysosome is less and it is acidic. It is rich in all the hydrolytic enzymes. And at low pH, all these hydrolytic enzymes are active. When they are released into the cytoplasm, they will digest all the carbohydrates, lipids, proteins present in the cell. Okay. What type of hydrolytic enzymes does lysosomes contain? Hydrolases, carbohydrases and lipases. Hydrolases also. Okay. So, if the pH is alkaline, what is the scenario is what he asked. Of course, if the pH is alkaline, hydrolytic enzymes will be inactive. That's it. It is as simple as it is. Okay. Hydrolytic enzymes will be active in the acidic pH. That is, it is when the pH is low. When pH is high or when it is alkaline, definitely those hydrolytic enzymes will be inactive. No, they will not be active. Option 1 is wrong. Hydrolytic enzymes will function more efficiently. Wrong. They will function efficiently only at low pH. Lysosomal enzymes will be released into the cytoplasm. No, they cannot be released. They are released only when they are in the active state. So, our answer is 3. Next question. Pathogenic bacteria gain resistance to antibiotics due to the changes in their what? Due to changes in their plasmid DNA. We have discussed, right? Plasmid DNA will imprint or it will give the particular phenotypic or specific phenotypic character to the bacteria. It will imprint what ma? Plasmids will imprint specific phenotypic character to bacteria. Okay. as they are giving out the specific phenotypic character to the bacteria. One such phenotypic characteristic feature is resistance to antibiotics, that is given by plasmid DNA, that is also called as extra chromosomal DNA. Next question. Here, this type of question we have discussed in APEAPCET also. Very important to learn the types or the shapes of the chromosomes, and you should also know in which stage or in which phase of cell division you can observe all the shapes of the chromosome. It is in the anaphase. It is in the anaphase of cell division you can observe the shapes of chromosomes. Shapes of chromosomes. Please fix it in your mind. Okay. Metacentric chromosome means centromere will be at the center and it has two equal arms of the chromatid. Means what will be the option? 3. Centromere is middle, forming two equal arms of the chromosomes, that is option 3. Next, acrocentric. Centrosome will be at one terminal end, forming one very short arm and one very long arm. So here what will be the option? Chromosome situated close to one forming end, that is option 1. Okay. Next, submetacentric chromosome means chromosome is close to one arm and far from one arm. Here the shorter arm of the chromatid both in submetacentric and acrocentric is called as P arm. It is called as what arm? P arm. And the longer arm is called as Q arm. It is called as what? Q arm. So here C is 4. Centromere is slightly away from the middle, forming one shorter arm and one longer arm. Here option E is option 4. Next, telocentric. Centromere is at the terminal end where the other arm is not visible only. You will find only one arm of the chromatid. Here the option will be 2. Then what will be our answer? A3, B1, C4 and D2. Option 3 is the correct answer. Next question of 2022 only, which is statement based. You see, it is regarding mycoplasma. Mycoplasma can pass through less than 1 micron filter paper. True, mycoplasma can pass through bacterial filter paper is the statement which is already in the NCERT. Mycoplasma can easily pass through bacterial filter paper. Bacterial filter paper. It is of what size? 1 micron filter paper size. Okay. And what is statement 2? The statement 1 is correct. 2 is mycoplasma are bacteria with the cell wall. No, mycoplasma are the only bacteria, they are without cell wall. They are without cell wall. Okay. So now what will be our answer? Statement 1 is true and 2 is false. Option 1 is the answer. Next question. Yes. Which of the following statements with respect to endoplasmic reticulum is incorrect? Okay, with respect to endoplasmic reticulum he is asking. In prokaryotes only RER is present. This is wrong because prokaryotes, just a minute, prokaryotes, they don't have membrane bound organelles. They don't have membrane bound organelles. In that case, how they will have RER, right? So this is wrong. Okay, they don't have membrane bound organelles, they don't have membrane bound nucleus, that is why they are called as prokaryotic cells. So they don't have RER. This statement 1 is wrong. SER synthesizes lipids and steroids. True. RER has ribosomes attached to endoplasmic reticulum. SER is devoid of ribosome. True. So yes, option 1 is our answer. Next question, if you see, when the centromere is situated in the middle with two equal arms, very simple question na, appeared in 2021. The answer is metacentric. Next question beta. Again, match the following question. One side the organelle is given, the other side its function and its location and shape is asked. If you see cristae are what? Infoldings of inner mitochondrial membrane. Infoldings of inner mitochondrial membrane. Option 3. Next, thylakoid, they are thylakoids are what? Flattened membranous sacs present in the stoma. Flattened membranous sacs present in the stoma. Stalk of this sac-like structures is called as grana. Stalk of this sac-like structure is called as what beta? Grana. So here B is option 4. Next, centromere. Centromere is what? Primary constriction of the chromosome, that is option 1. Next, cisternae, they are disc shaped sacs in the Golgi apparatus. They are disc shaped sacs in the Golgi apparatus, option 2. So if you look into the proper sequence of the options, A is 3, B 4, C 1 and D 2, option 4 is our answer. Next question beta. When the centromere is situated in the middle of two equal arms, then the chromosome is referred as. See how many times this question is asked based on the shapes of the chromosome and particularly metacentric chromosome. The answer, when two equal arms are given, then our answer will be what ma? Metacentric chromosome. The organelles that are included in the endomembrane system, see n number of times endomembrane system also repeated, that is what? Endoplasmic reticulum, Golgi bodies, lysosomes and vacuoles. Option 3 is our answer. Next question. If you look over here, which is the important site for glycoproteins and glycolipids in eukaryotic cell? Site for the formation of glycoproteins, glycolipids, obviously it is Golgi bodies, asked in 2020. Next, which of the following is important site for the formation of glycoproteins and glycolipids? Same question, actually it is repeated. Next, inclusion bodies of blue, green, purple and green photosynthetic bacteria. What are these inclusion bodies? Inclusion bodies are non-membrane bound organelles which are present in the prokaryotic cells. What are these beta? Non-membrane bound organelles. Non-membrane bound organelles which are present in the cytoplasm of prokaryotic cell. These organelles, what is their function? They store reserve food material. They store reserve food material. They store reserve food material. Okay. So here what will be our answer? Inclusion bodies in blue, green, purple and green photosynthetic bacteria are nothing but gas vacuoles. This is of course the statement from NCERT only. Next. Match the following columns and select the correct option. See, again match the following questions. Of course, we have discussed this in our match the following session also. Smooth endoplasmic reticulum, of course 2, lipid synthesis. Rough endoplasmic reticulum, protein synthesis. Golgi complex, 3, glycosylation. And third it is centriole beta. Third it is what? Centriole spindle formation, that is 4. Okay, if you look into the options, it is 2. Smooth endoplasmic reticulum, it is 2. I think it is not given. Please look over here. A is 2, B is 1, C is 3, C is 3, D is 4. Glycosylation is nothing but formation of glycolipids and glycoproteins. Packing or packaging of proteins and lipids in the Golgi by adding sugars like glucose, mannose, fructose and making them or packing them into glycolipids and glycoproteins is called as glycosylation that happens in Golgi bodies. This is the sequence of the option. Somewhere it is mistaken over here. Next. Yeah. Size of pleuropneumonia like organisms he has asked. In the diagram where he has mentioned in the NCERT that different sizes of the cells, in that he has mentioned the size of pleuropneumonia like organisms, that is 0.1 micrometers. 0.02 to 0.2 micrometers is for virus. This is the size of what beta? Virus, approximate size of virus. 1 to 2 micrometers is the approximate size of bacteria. It is of bacteria. 10 to 20 micrometer size is of what ma? Eukaryotes, eukaryotic cell. 10 to 20 micrometer size of cell is of what ma? Eukaryotic cell. Next question, if you see, which of the following cell organelles is present in the highest number in the secretory cells? Highest number of the cell or these organelles are largely present in the secretory cells. For example, root cap region, cells in root cap region, cells in root cap region, cells of acrosome of sperm, acrosome of sperm. This region, this region acrosome of sperm, it is also rich in Golgi complex, Golgi complex or Golgi bodies, where it will secrete all the secretory substances like glycolipids, glycoproteins and all the molecules, okay, which are sticky in nature, that will provide the positively geotrophic movement of the root region. Okay, so these are secretory cells, examples of few secretory cells wherein you will find which type of organelles beta? Golgi bodies, because they will secrete lot of the molecules which are packed from lipids and proteins. Okay, next. Yes, non-membranous nucleoplasmic structures in the nucleus are the site for the active synthesis of. This question, if you remember, in the beginning of this session we have discussed about this question, but in a different pattern. Non-membranous nucleoplasmic structures are what ma? Nucleolus. Non-membranous nucleoplasmic structure are nucleolus. Nucleolus is involved in the synthesis of rRNA. Okay, and if you see nucleoplasm is involved in the synthesis of tRNA and mRNA. So here the answer will be non-membranous nucleoplasmic structures of the nucleolus will synthesize what? rRNA, ribosomal RNA. Next question, if you see, the shorter and longer arms of submetacentric chromosome. Shorter arm we will call submetacentric chromosome, where centromere is slightly away from the arms of the chromatid, where in the shorter arm you will call it as P arm, longer arm you will call it as what beta? Q arm. Okay, so yes, shorter is P arm and longer is Q arm. Option 2 is the correct answer. Next question. Which of the following statements is not correct? Okay, lysosomes have numerous hydrolytic enzymes. Of course, that is a true statement. The hydrolytic enzymes of lysosomes are active under acidic pH. Yes, exactly correct. Lysosomes are membrane bound structures. True. Lysosomes are formed by packaging in endoplasmic reticulum. Wrong. They are formed by packaging in Golgi bodies. They are formed by packaging in what ma? Golgi bodies. Yes. So this is the wrong statement. Our answer will be wrong. So check it properly whether he is asking incorrect statement or correct statement. Okay. Then you go for the options. Which of the following statements regarding mitochondria is incorrect? Mitochondria, if you see, outer membrane is permeable to monomers of carbohydrates, fats and proteins. True, correct. Outer mitochondrial membrane, yes, here I will draw. Outer mitochondrial membrane is permeable for everything. Inner mitochondrial membrane has enfoldings. You will call it as cristae. Enzymes of ETS are embedded in the outer membrane. Wrong. All the enzymes in inner mitochondrial membrane, all the enzymes, electron carriers and channels required for the transport are present in inner mitochondrial membrane only, not in the outer mitochondrial membrane. So yes, 2 is wrong. Inner mitochondrial membrane is convoluted with enfoldings called as cristae. True. Convoluted with enfoldings, then it is true. Mitochondrial matrix contain single circular DNA molecule. Yes, single circular he said, don't get confused. He didn't say single stranded, okay. He said single circular DNA. Then and ribosomes, nuclear envelope and mitochondria. Correct only. Yes, clear. Next, so what is the option we need to choose? He asked us again incorrect statement. So the option is 2 only. Clear? Next question. The concept of omnis cellula e cellulae regarding cell division was proposed by Rudolf Virchow, where he mentioned that all cells arise from pre-existing cells in the year 1855. Next question. Many ribosomes may associate with single mRNA to form multiple copies of polypeptide simultaneously. You will see this in the gene expression under mol bio. For example, Lacoperon model you will see, wherein single mRNA chain from 5 prime to 3 prime. Single mRNA chain is attached with many ribosomal subunits. Single mRNA chain is attached with what beta? Many ribosomal subunits, wherein it can synthesize polypeptide or wherein it can form polypeptide chain. Polypeptide chain, which is nothing but the multiple protein expression. Multiple protein expression you will call it as what ma? Polycystron. Polycystronic expression is seen. Polycystronic expression. Okay. For example, that is why you will see 3 structural genes na in Lacoperon, that is Z, Y, A. Each is coding for one enzyme. Clear? So yes, that happens only in the prokaryotic cell, and the pattern wherein in single mRNA multiple ribosomes are attached, that model you will call it as polysome. Polysome. So yes, the option is 1. Many ribosomes associated with single mRNA, that condition is called as what ma? Polysome. Polysome you will see in a prokaryotic cell. You will see in what ma? Prokaryotic cell. Monosome, wherein single mRNA is associated with only one ribosome, that is called as what ma? Monosome. That is called as monosome. This you will see in a eukaryotic cell. This you will see in what beta? Eukaryotic cell. Next question, if you observe, next question. Which among the following is not a prokaryote? If you look into it keenly, all of them are unicellular, but not all of them are prokaryotes. Saccharomyces cerevisiae is a unicellular fungi. Belongs to what class of fungi ma? Ascomycetes. They belongs to Ascomycetes, which is also called as yeast or baker's yeast. Okay. Next, Mycobacterium belongs to bacteria. Mycobacterium belongs to bacteria. Oscillatoria and nostoc are what? Cyanobacteria. They are what? Cyanobacteria. Both of them are prokaryotic. They are what? Prokaryotic. They don't have membrane bound organelles or membrane bound nucleus. So our answer is 1. Next, the Golgi complex participates in formation of secretory vesicles, like lysosomes, glycolipids, glycoproteins. All these are liberated out or released out of the cell in the form of vesicles from what? Golgi bodies. Next question. Next, that is asked in the year 2018. Which of the following is true for nucleolus? He is asking. Nucleolus is what? Non-membrane bound structure of the nucleus. See, this is third time the question is repeated. Next, which of the following is true for nucleolus? He is asking. Larger nucleoli are present in dividing cell. It is membrane bound structure. This is wrong. It is site for rRNA synthesis. This is true. See, nucleolus question only with respect to rRNA. This is third time we are looking into this question. Okay. Next, which of the following cell organelles is responsible for extracting energy from carbohydrates to proteins? See, preparation of complex carbohydrates, the activity is done in chloroplast if we consider plant cell. But then the energy required for all the metabolic activities of the living organism or the living cell is given from mitochondria in the form of ATP. The cellular energy is created from the food whichever we take. That activity happens in ATP only. Finally, it will release what? Cellular energy from whatever food we take. Okay, this oxidation of macromolecules starts from cytoplasm but happens in mitochondria in a eukaryotic cell, wherein finally it releases ATP, wherein this is used for all the metabolic activities of a cell. Next question. Which of the following are the components, which of the following components provide sticky character to the bacterial cell? It is glycocalyx. Glycocalyx will produce the sticky character to the bacterial cell. Glycocalyx can exist in two forms. Glycocalyx can exist in two forms. One form is capsule and the other form is slime layer. Slime layer. In glycocalyx, if the molecules, all the molecules are close to each other, then they form a thick capsule. This capsule helps the bacteria to hide from host immune system. To hide from host immune system. Host immune system. Slime layer, they are loosely arranged. All the molecules which are present in the glycocalyx, they are loosely arranged in the slime layer, wherein this prevents the bacteria. This prevents the bacteria from loss of minerals and nutrients. Simply dehydration. It will not allow the bacteria to dehydrate if it is in the form of slime layer. If it is in the form of capsule, it will tightly uncover the bacteria and it will prevent the bacteria and it will help the bacteria to hide from host immune system. This is all about glycocalyx. Next question appeared in 2016. Select the mismatch. Okay, gas vacuoles in green bacteria, large central vacuoles in animal cell. Just by looking into it, you can strike it off, because large central vacuoles are present in plant cells as they cannot move. All the excretory products and sap of each cell is stored in vacuole. That is why vacuole in a plant cell will occupy 90% of the space of the plant cell. Okay. Next question. Next question, if you see, yes, a cell organelle containing hydrolytic enzymes are lysosomes. Of course, they are what ma? Lysosomes, which are of hydrolytic activity. Water soluble pigments in a plant cell vacuole. What actually the pigments are of two types. What are they? Pigments are of two types. One is fat soluble, one is fat soluble, and the other are water soluble pigments. Water soluble pigments. Fat soluble pigments are carotenoids, xanthophylls, and chlorophyll. Water soluble are phycobilins. For example, phycocyanin and phycoerythrin and anthocyanin. These are flavonoid derivatives. They are flavonoid derivatives. Okay? Yes. So, what will be our answer over here? Water soluble pigments he is asking, found in plant cells vacuole. They are what ma? Anthocyanins. All these three are what beta? Fat soluble pigments. Next question. Mitochondria and chloroplast are semi-autonomous organelle. True. A is correct. A is correct. This is the last question we are exercising today. Yes, they are semi-autonomous or self-replicating organelles which have their own DNA, that is double-stranded circular DNA. Formed by division of pre-existing cells, yes, they contain DNA, true, but they lack protein synthesizing machinery. No, they do have protein synthesizing machinery, and the rubisco, that is the world's abundant protein synthesized by the double-stranded circular DNA which is present in the stroma of chloroplast. So, B is wrong. Then what will be our answer? Mitochondria and chloroplast, state both, A is true and B is false. This is all for today. All the PYQs of NEET are very, very important. If you revise it and go, you will cover so many concepts of cell the unit of life. So, don't miss this session. All the best for your exam. Thank you.